Okay, good afternoon, and thank you all for being here today. Welcome to this virtual space convened by UC Berkeley's Cannabis Research Center. My name is Joanna Hossack. I am a specialist in the Institute of Urban and Regional Development. For those of you who don't know, the Cannabis Research Center has been around since 2017. And since then, we've been studying issues related to cannabis agriculture, the environment, and local land use policy. I invite you to check out our website, crc.berkeley.edu, to view our past webinars and future events and to read our reports. You can also sign up for our newsletter. We're going to include a link in the chat. Today, we're very excited to launch our California Research Conversation Series, where we will interview California cannabis and hemp researchers about their work and invite our audience to ask questions and explore these topics in depth. The conversation series will take place virtually every other Friday throughout the fall and spring, and we're going to include our link tree in the chat so you can register for future events. We are thrilled to welcome our speaker, Dr. Thomas Azwell, who is a UC Berkeley faculty member and director of the Disaster Lab. We're also going to share a link to that in the chat. And he is here today to discuss his research entitled Variation Among Hemp Analytical Testing Lab Evidences Regulatory and Quality Control Issues for the Industry, which was published in the Journal of Applied Research on Medicinal and Aromatic Plants in December 2022. We're also going to include a link to that report in the chat so you can take a look at it. Before we jump in, we'll let you know about the format of this meeting. This discussion will last for 1 hour, ending promptly at 1 p.m. We will start with an overview of the research, followed by prepared questions, and then at the end, we'll open it up to the audience Q&A. Please remain on mute unless you are prompted to ask a question. As we move through the discussion, please put your questions in the chat. When we get to the Q&A portion, I will select questions to be answered, and you can unmute to ask your question. Please note that this meeting is being recorded, and it will be posted to our website later. So let's begin. Dr. Azwell, thank you so much for being here. Could you please tell us about your current role at UC Berkeley and how you came to work on hemp research?
Sure, thank you. So I am actually a graduate of UC Berkeley. I graduated from the College of Natural Resources. My initial research was on organic agriculture, and then the Gulf oil spill happened. And I got distracted and ended up working on an environmental impact report for the BP oil spill. And so I had this agricultural interest and background that has stayed with me even to today. But we ended up getting most of our opportunities in working on large scale natural disasters or human caused disasters like oil spills, and more recently in California with wildfires. So that was the impetus for the Disaster Lab. And that was and it made sense with looking at technologies to help mitigate environmental disasters, that we were to move from College of Natural Resources to College of Engineering, where innovations are built. And then our kind of main role is in this whole process is really helping to facilitate adoption of innovation, which is turning out to be the hardest part. So there's lots of technologies and innovations that could be used that are available or being developed by the industry for disaster and environmental mitigation, response, and restoration, but aren't utilized. And so that's a major role that we play in the field. And then I think the beginning of your question was, how did I get into hemp? We just accidentally, I had an alum that was making an investment in the space, and this was right after the Farm Bill was passed, that legalized hemp, that took it off of its Schedule A classification. And he wanted us to advise the conversion of a 5,000 acre alfalfa farm in Utah to hemp. So our lab group started looking at, the state of the industry, and it was really the Wild West back then. It was a lot of folks that had done work in cannabis agriculture, and a lot of those representatives had been doing it, below ground, and they were coming out as kind of consultants. There was also a lot of demand for high CBD seed, and then what was the value of those seeds were represented by what's called a certificate of analysis. This was a lab result, not on the seed itself, but of the potential flower that would be harvested from that seed. And so we started asking questions about, how representative was that, and really got interested in how we could support farmers in this process of becoming hemp farmers for the first time. A lot of them kind of betting their farms on this new boondoggle of an industry, which was high CBD hemp. So that's how we got involved.
Amazing, really pioneering here. It's a very emerging issue, so thank you so much for sharing. You please provide a brief overview of the research we'll be discussing today.
So the most recent publication that we did was the result of us working with commercial regulatory labs as a way to kind of ground truth our testing that we were doing in our lab. So just a third-party analysis, or working with these the same labs that were using the same equipment that we were using in our lab. We'd purchased an HPLC. Shimadzu was a company that was manufacturing those, and they had put a hemp leaf on it and called it a hemp analyzer. And they were probably selling more HPLCs than anyone as a result of that. So it was a great marketing tool. But what we found out really quickly when we purchased it was they had a hard time kind of supporting us in not its use, but its calibration for hemp. So I'm not a lab scientist, but I work with folks that are, and this is part of the great thing about being at Berkeley is there's experts in every field just out my door that I can go and talk to and rely on for doing these things. So I worked with folks that ran labs on campus, that ran HPLCs and did chromatography. And one of the things I learned is that an HPLC is not exactly the best tool for measuring a resin-based compound like cannabinoids. It is hard to calibrate and to get a very accurate result. And when I say accurate, I'm referring specifically to the hemp industry's regulatory standard, which is that the hemp flower has to test at 0.3%, 2 weeks, sometimes 4, sometimes it changes to 2 prior to harvest. And that's a tricky thing for a farmer. And so we met a lot of farmers that had failed that test prior to harvest because they were waiting to call the county regulators until they were trying to push their crop as far as they could. And so rather than harvesting early, they were making that phone call to the ag commissioner to do the inspection 2 weeks prior to when they were really going to harvest. But what happens with hemp and same with cannabis is the levels of cannabinoids spike towards the end of the season during the flowering process. So you have this exponential increase in CBD, but you're also getting an increase in THC at the same time. So they were trying to push the time to harvest, but at the same time, they were also pushing the risk of not being compliant and having a crop which they called going hot. And so depending on that test result and depending on that lab that did that test and when they did it, that test result could end in either someone from the DEA being contacted to come and watch you destroy your crop, to the local ag commissioner just saying you have to mow it or till it into the ground, or to cut the plants and burn it, to maybe just a slap on the hand. And so that was we were seeing farmers that had spent upwards of $1 million to get that crop to harvest, that were then being told that that their crop had gone hot and that they have to destroy the entire crop. So the first thing that we did to start supporting farmers is worked on the genetics. And so we built a selective breeding program that utilized high throughput screening, and a very specific process that helps to stabilize the genetics so that you don't get what they call phenotypic drift between plants of a certain cultivar. Because as we know, if you take two different plants and you breed them, the children of those plants are not going to be exactly the same as the parents. And there'll be variation amongst the children between the sisters of those parents, and that variation could be the results of testing hot because the ag commissioner came out to pull some flower for the sampling, and happened to choose some of the plants that were exhibiting the highest levels of THC. So that's where we started kind of addressing those pain points, trying to do it both from a breeding perspective, but also getting really interested in the policy. And it's what led to us even thinking about doing that paper, which was, let's send multiple regulatory labs the exact same flower, and replicates of the same flower, and look at the results. And these are regulatory labs that are being utilized to hold hemp farmers to compliance, all testing the same flower, not knowing that they're testing the same flower, and multiple samples of the same flower that we labeled differently, and all coming back with different results, which that was our hypothesis because we had experienced that. We'd experienced it in our own lab. So we knew it was going to happen in a regulatory lab, probably even more so, because of the throughput that they have running samples. They're not taking the time to calibrate as often, and they're not their instrument, and they're not taking the time to clean their columns between samples.
And I think it was 13 labs that you worked with.
I think so. Yeah. I think
Yeah.
13 labs that weren't going to charge us the full price, and we could get an academic rate. All of the research that we did was almost all of it was unsponsored. This is research that we just funded ourselves. So we were on a budget for all of this. There was not a lot of grant money out there. Most of the grant money was really to look at addiction, or maybe some medical applications of cannabinoids. If you're looking at hemp with glaucoma and other diseases. But there wasn't a lot to look at policy, regulation, genetics, and the like. So we were on a budget. So we found labs that we'd worked with before, and others that would work with us on a budget to do that testing. And one of the labs was one that we work with on a regular basis. And I just have one really quick story about that lab. This is a California-based lab that's used by most ag commissioners to do regulatory testing. well-established lab. As part of my research in the medical field with the University of Chicago on COVID, we ended up sending them a synthetic CBD. And I got a test result, a COA, a certificate of analysis that had THC in it. And I quickly called them up and said, that's not possible, because what I sent you was a synthate. It's a synthesized CBD. It didn't come from a plant. It's not possible. And they said, we'll retest it. Don't worry. They retested it, and a few hours later, I received an updated COA that didn't have THC in it.
Oh, wow.
They were surprised that I called because the THC result was below the 3% threshold.
Mm-hmm.
And they thought that was what I was looking for. And I wasn't. I was just looking for an actual result, whatever their instrument tested it at. So I can only speculate what happened in that scenario, but just from our own best practices in our lab. So I don't want to, hypothesize, but I just know from our experience that if you're not calibrating your instrument, which costs upwards of $1,100 to do a calibration, then, and you're not cleaning your column, which it's hard to clean with a resin-based compound that you're running through it, then it's you're not going to get consistency in your test results.
Okay, wow. So before we get into the lack of standardization, can we talk a little bit about sampling, just to give the audience an idea of what that looks like? So, what kind of process did you go through to send a sample to the lab, and what kind of issues arise during the sampling and just getting it to the lab, that process?
Great. So I'm gonna talk from the entire kind of community of people that sample and send for testing, just to give you that perspective, because I think the most interesting thing that I learned from this experience was that we got to sit in the front row of watching an investor invest vertically in the hemp industry, and all of the challenges that they experience. So that was what we learned from this. What our perspective is seeing where the challenges were. If you look vertically in that industry, and vertically meaning you start from the person who's producing the seed, to the farmer that is planting it, to the extraction lab that is doing the extraction to prepare the product, to the product manufacturers. So that whole supply to the consumer, to my mom's friends who call me up and say, what brand of CBD should I be buying for my pain cream? So that is the perspective that I have. The farmers are harvesting themselves to do their own testing on their genetics. They're usually pulling kind of a representative sample, which would be not the biggest plant and not necessarily the top flower. It's going to be somewhere in the middle, and then that's going to give them kind of a good average if they grab from about a dozen plants in different parts of the field. And so they're cutting the flower, putting it in a Ziploc, and shipping it to a lab. All the way from the regulators, who have their own process for how they're going to collect samples. The empathetic ones are going to collect bottom flower. They're going to grab a lot of stem and leaves in that sample in hopes that the farmer will pass the test. And that is how samples are, and again, being just put into a Ziploc and a FedEx package or whatever shipping mechanism that you have, that there's not scrutiny, because FedEx can sometimes can scrutinize what's in their packages versus UPS. So there are differences in who the shippers are. that gets to the regulatory lab. The lab will then go and do an extraction, and they'll use the solvent of their choice. A lot of them use methanol because it's cheaper. Yeah, we use ethanol because it's less toxic. If we're going to prepare something for medicinal testing, we'll actually use ether because you don't have the residuals. You can volatilize it off, and also you're not pulling out, like if you're a plant biologist, you know that when you do extractions with methanol or ethanol, you're just simply lysing the cells and everything comes out. You get the paraffin waxes and chlorophyll and everything gets in your sample. So that's how they're just doing a quick extraction and then running it on their column. So that's the process where the variances in that whole process is things like heat and sunlight and time can cause degradation. So it may not, and all the things I mentioned earlier about the testing can cause that variation in that result.
Wow. So there's a lot that could go wrong just during the sampling before we even get to the next step.
Correct.
Wow. Okay. Thank you for explaining that to me. I did not know that process. It's always been a mystery. So how would you suggest addressing the lack of standardization or standardized testing protocols among hemp testing labs to ensure that they are having a more reliable THC result?
Yeah, so what I did when I learned of these problems and wanted to try to affect some change to support the industry and farmers and create some standardization is one is I wanted to move suggested moving away from HPLCs and using a different tool and a different instrument for testing. I mean, the idea of testing is proving that it's hemp, not cannabis, that you're growing. I mean, that's what you're supposed to be doing. The 0.3% is flawed because all high CBD hemp is going to finish. When I mean finish, if you take it all the way as far as it'll grow before it starts to degrade, it will test at over 0.3%. It'll test as high as 0.8 and maybe even 1%. So
wow.
Part of the struggle with breeding high CBD hemp is you're also going to be breeding high THC hemp. And high meaning less than 1% at harvest. But if the regulatory limit is 0.3%, based on a 1977 publication by a botanist in Canada, who was just differentiating between cannabis and industrial hemp at the time. His name is Ernest Small. I talked to him because I was like, what do you think about the fact that they adopted your 0.3%, botanical classification as a regulatory tool? And he said, he goes, I'm flattered they read my paper so long ago and everybody's talking about it, but that's not what it was intended for. And basically what we talked about was, what is the purpose of that regulation? And the purpose is to allow farmers to grow hemp under that regulation, but not cannabis.
Right.
And if you are a cannabis farmer or a hemp farmer and you're trying to sneak in cannabis and your cannabis is 0.8% THC, then you're probably the worst cannabis farmer in the world. So the fact that would even be in question. So what Ernest and I talked about was really, where does the purpose of the regulation is to ensure that what farmers are growing is not cannabis, so that so it can be sold as a CBD product and not have that psychotropic effect. And it's got to be over 0.8% before you, as a consumer, before you actually can experience a psychotropic effect.
Okay.
And so our suggestion was, why not just change the regulation to 0.8% or higher? And then I would say 95% of the farmers would be compliant. 95% of the farmers would be compliant 100% of the time. If and it would kind of take that pressure away from the regulators, the regulatory labs, and the farmers themselves of when do they make that phone call to get that inspection to see if they're compliant and they're not going to lose their farm on that year's crop, because the farmer themselves did everything that they could to ensure that they were buying hemp seed. They bought hemp seeds from somebody that was certified to sell them, that came with a certificate of analysis that showed that they were below 0.3% THC. Well, those seeds weren't, which is the other problem. It was whatever produced those seeds, if that was even what was tested to get that certificate. It wasn't those seeds. The person selling the seeds didn't go and produce seeds and then take a handful of those and grow them to harvest, and then send the resulting flower into a lab to get a certificate of analysis. They didn't do that because that would have taken a year. It would have taken a season at least of growing and harvesting. And so I know that's not, those certificates don't represent those seeds. They may represent those seeds are the progeny of whatever, potentially of what that COA was.
Wow, I didn't know that. So even where you got your seeds, that person, it was relying on test results that may, right, not.
Which are not those seeds. They were relying
Right.
on test results that showed high CBD and low THC because that was the value of the seed. The value of the seed was being compliant below the 0.3%, but being as high CBD as you could get.
Right.
And these are PDF documents that can be fudged. Not all of them have QR codes that go to the lab's test result site.
Wow.
And so, and at the beginning of the hemp industry, the 2018 industry, those seeds were being sold for north of $1 a seed. That's what it was. That's what the market was bringing. I remember being on a phone call with that investor that was going to plant his farm, talking to a seed grower who was at the Olympics in France, calling from the stands, not even stepping out because he had so much demand. He was making so much money. He was actually at the Olympics talking. You could hear the crowd yelling in the background, and he was talking. So you want to buy my seed or what? It's going to be $1.50 a seed. That's what it's going to be. Didn't you see our COAs?
Wow.
That was the Wild West that I was referencing.
I didn't realize how much of a minefield it is and how just you're walking a tightrope there.
Yeah, so I think it's changed a lot. A lot of that uncertainty or that the kind of the industry itself is becoming more standardized, more less people are throwing funny money at it, and there's less investment. I know a lot of that venture capital that went into it were venture capitalists that couldn't invest in cannabis because their charters didn't allow them. And hemp was an opportunity for them to invest in something cool.
Right.
And so they were throwing a lot of money in it. What I ended up doing with that conversation with Ernest Small was I was having a lunch and learn with the USDA and all the folks that are responsible for the policy around hemp and that understand this problem really well, and talking to them about alternatives to the 0.3%, the use of HPLCs, the variation that we see in the lab, the standard deviation that we see in the lab that can throw off a result. And I really did think that I debated on this, but that paper that I published could have been used by all those farmers in a class action lawsuit that had to destroy their crops to go back and say, wait a second, How do we know that that lab that you used was calibrating their instrument and we're getting an accurate result? Look at this publication that shows this variation. When you're looking at something at such a minute amount, and you have that level of deviation, they could have gone back and sued for. And so I thought that might be the result of that publication, but it wasn't. But one of the things I suggested to the USDA is maybe looking at doing a genetic analysis instead, because if you do a genetic analysis on the difference between hemp and cannabis, you actually see the difference is in the THC-A allele. So one of the alleles in the THC, so hemp can still produce THC, but it can never do it at the levels because of that genetic difference. Why not do a genetic analysis? Or why not regulate it at the extraction facility, beyond the farmer, to that next stage of the industry, and make sure what's coming out of the extraction facility that's going into the products is compliant, and not putting the onus on the farmer, or just changing the level to 0.8%, and Bob's your uncle. And they all agreed with that assessment, but they all kind of knew that that type of change is going to take a while. So I think there's still a lot of debate. The response that that ended up happening as a result of these conversations was that NIST, the standardization regulators, put out what they call reference samples. So these labs are now being, I think it's suggested, but it's going to be mandated that they need to calibrate against these reference samples that you can buy to make sure that your instrument is getting the same result as the certificate that comes with those reference samples. But again, those are flower, and I didn't fully answer your question. The way we prep our flower for the HPLC is we use a very expensive blender called a Geno grinder that pulverizes using metal balls the flower to make sure that we get a full extraction. So we get a very accurate reading, so we get the same result that the extraction facility, the industrial extraction facility, would get on the other end of their extraction process. Most labs don't invest in a blender. They just do it in a petri dish or a pestle mortar. A pestle mortar with some ethanol or methanol and get an extract. So we were very thorough in the extraction, but most labs aren't that thorough. They're not removing the stem and all of those other things. So again, leading to that variation. Sorry, I'm going on.
No, this is great. This is wonderful. I'm just going to remind everyone that if you have any questions, please go ahead and put them in the chat. We're going to move to audience questions in a minute. We just have a couple more questions. So, I mean, we're kind of weaving through a few different questions here, but you mentioned the USDA, meeting with the USDA, but what role should regulatory agencies play in standardizing reference materials and calibration methods to reduce lab variability? And you've kind of touched on this already, but.
Yeah, and so I think that if they, I understand why the regulation was created, understand the intent of it, but I also understand how it's flawed and all of the reasons why. The regulators understand this too, and you can see it when they come out to a field and they harvest. I can tell, like I was saying, if they're empathetic or not. There's very few ag commissioners that have their own labs. Utah had their own. They used their own lab. They were doing a lot of testing. A lot of these ag commissioners have testing labs that were set up for pesticide, for the organic industry, really to look for residuals and pesticides and things in organic crops to test against that. So they did have these chemistry labs. Utah is the only one that I came across that was actually using their own lab.
Mm.
And we spent a lot of time talking to the person running it. So I think that they should be inspecting the commercial labs that they're using for regulation. If they're going to, the onus should be on their regulators to make sure that the labs that they're collaborating with are putting out a representative result,
Mm. Mhm.
not on the farmer.
Right, right. To the ag commissioners and the agricultural departments across different states. Okay. So we have one audience question that we can go to for now, and then we might go back to some of the questions I had prepared, which is, do you participate with ASTM to put some of your own findings into standardization for labs and testing?
I think that I don't, but I think that would be, the next iteration of our research if we were to continue that research. I didn't mention all the reasons why we don't do this research anymore, but I think it's important.
Mhm.
And I remember the first time that I attended a CRC meeting years ago and talked about the challenges I was experiencing, even doing research in this field. And I think as a group, the consensus was, well, maybe you shouldn't associate yourself with cannabis if it's the stigma of cannabis that's preventing you from doing research in hemp.
Mm.
So the last time I tried, I wasn't able to grow hemp on campus. So all of our research has to be done off campus. And it is a real challenge to do, and I'm sure it's even more so in the cannabis and psilocybin and these other, medicinal industries that aren't really treated as medicines yet, or not really, that have this stigma, because how they've historically been used, culturally or
Mm.
medicinally as as not a typical drug that's developed through the typical synthetic process. So a lot of the reasons why we stopped was the lack of funding, the stigma, and just the critique and oversight that we got, and the pushback that we got from everyone, both in academia and beyond, of even being affiliated with this industry. And I think of this one moment that was really stark for me. There was a panel discussion in Australia, in Melbourne, and Melbourne was kind of leading the way of the hemp industry growth in Australia, and I attended this. And there was an audience of academics and industry reps and all kinds of folks, and there was a panel of real serious people that were, and I say serious in that there is medicinal value in these constituents of these plants, and it's you can find anywhere from peer-reviewed publications to anecdotal discussions with people that have been taking it. I've made a pain cream from our leftovers and given it out, and I live next door to 4 nuns that are elderly. They teach at the theology school, and they've been using it for arthritis. One had a frozen joint, and every time she sees me, she points at me now because
I love that.
and that was where I decided that if I'm going to be, I'm not, if I'm going to have a career in academia, that I need to choose a different topic to work on. And that's where, I don't get that same response when I'm working on disasters and technologies related to that. But I think that the ASTM question is a really good next iteration of where this work should go, and really just flesh out the fact that the current instruments that are being used just cannot get that type of accuracy at that low percentage. And so for the cannabis industry, if you have a difference of 0.5% plus or minus on THC, when you're talking about a 20% product, it's not going to be noticeable or consequential. Maybe a little bit on the pricing, but not much on on the industry side from what I discussed. But with hemp, it's the beginning to the end.
Right.
So
To
it's
make or
definitely
break.
something that needs to happen. And I wish, UC Berkeley and some of the academic community was able to kind of lead that effort, and there was funding to do it.
Mhm. I am optimistic that as cannabis is liberalized nationally and globally, and the stigma slowly starts to fade away, one day there will be more opportunities. So hopefully that day will come sooner than we expect. So how do you think we could revise existing hemp regulations to reduce the risk of farmers not being in compliance the weeks prior to harvest? You've mentioned changing the percentage. Are there any other revisions?
Yeah, I mean, I think if you just regulated the seed and were able to just say this is hemp seed, not cannabis, that's good enough right there. Having to go out to a farm and do an inspection, everybody's harvesting close to the same time. It's a really stressful part of the process of growing hemp, and knowing when that is and when to harvest, especially when there's value in things like terpenes. If you are harvesting late in the day, a lot of your terpenes have volatilized off for the day. It's just there's not a lot of farmers that have this kind of generational knowledge of growing hemp,
Right.
or growing cannabis. So they are taking a lot of risks. So if you can remove that risk through one of the ways that I mentioned, and just shifting that regulation to a different part of that supply chain, I think that would be one way to allow the smaller farmers to participate in the industry, and not fast track this to big ag, or outsource it, or where it's going to end up in other countries where there may not be so much regulation around the use of pesticides and
So true.
other things. So
you also mentioned perhaps moving this to the manufacturing level. Can you describe that a little bit more?
sure. So I didn't mention the challenge with extraction facilities, but the last time I worked with an extraction facility and a farmer on getting the product converted for sale, that extraction facility was going to charge the farmer 50% of their yield or more to do that service. Rather than just giving them a price, they say, we're going to take 50% of your yield. The farmer not knowing what the real yield is, and just saying, okay, what else can I do? The more I let it sit around, it's going to degrade. There are 3 seasons of crops sitting at in Sonoma at an academic institution there, that they couldn't afford or didn't find a lab to do, or a extraction facility to do the extraction for them. They even asked me if I wanted it, and it's all moldy because it wasn't stored right, or so it's unfortunate. But the extraction facilities who do that conversion to get a saleable product, whether it's just a full spectrum extract, or they're converting it to an isolate, or other product that can then be sold, so the farmer can get that revenue on that end. I think that's where a lot of this can happen. I was interested in how the regulatory, or how the extractors could go and remove the THC, but what I've learned is when they do that step, they also remove some of the beneficial properties of a full spec product, when they're making what they call THC free extract. And so if it's not going to be used for medicinal purposes, I'm not really sure. I mean, it needs, there needs to be more focus on that step. If you're really going to be selling this as a medicinal product, whether it's to help you with anxiety or sleep, or some of the other things that I mentioned as a topical, or even for use with COVID. What we found is we bought CBD isolate as part of the University of Chicago research from suppliers all over the United States. And what we found is none of the CBD isolate that we purchased was more than 95% pure CBD. And that was an interesting result. If you're buying stuff that's being sold as 99.9% pure CBD isolate, and it's 95% at best, how can you use that in medical research?
Right.
If you have that uncertainty of what those other constituents are that are going to lead to it. The only 99.9% CBD isolate that we purchased that we found was actually synthetic, that was being sold as a plant-derived isolate. So we discovered that synthetic in the industry, and it was effective in the research that we did on the animal studies for COVID. The synthate works on that application. And the final result of that research was the recommendation from the pharmaceutical industry that they do use a synthate, which is probably what they would have done if they actually had gone to market with the drug. But when the University of Chicago went to get to a clinical trial, their university didn't support them. And this is a group of PhD MDs, 16 of them that are part of a medical school. And their development office for the University of Chicago, because they're a land-grant university like UC Berkeley, said, we're nervous about being involved in this type of research, and we don't want to support you going to a clinical trial.
What a shame.
So that COVID application, which it kind of, it does have potential. What it does with COVID, if taken properly in the right form and the right amount, and you can see this in their publication, some references to it, it actually elicits an immune response early on when you're infected, which is where COVID really has been able to become a pandemic, is it suppresses your immune response, allows itself to establish and replicate. And then by the time your body responds to it, you're already ill.
Wow.
What we found with CBD, if taken right, that it can actually help your immune system elicit an immune response sooner.
Wow.
So much potential there. And we found this through animal studies, self-funded research, and discovered some new things about the industry with the CBD isolate being sold. And again, we were stopped from proceeding. But the most interesting thing to me was that lead researcher, who's a senior faculty member at University of Chicago, was just shocked by the press attention that she got from that study, that publication in Nature, but also the questions that she got over and over again about, can we just smoke some weed? Is that going to help us from getting COVID? And she was surprised. I mean, she's still shocked today that, she spent 90% of those conversations talking about the difference between cannabis and hemp.
Mm,
and how this would work as a drug, and just getting
right.
past that
Right.
those questions and stigmas and misunderstandings. Or yeah.
Wow, that is unfortunate that it went down that way because that could have helped a lot of people. So just so to recap, that is synthetic CBD was helpful at creating that immune response. And do you recall what the dosage was or like what the
I'd have to look back to my notes. But our in plant-derived CBD would have worked just
okay.
it's just they ended up using the synthate for the studies in order to reduce those confounding variables, is how
Yep.
it's described, because of those other ingredients, or those unknowns. But if you take it in a high dose, and you take it in a form that it doesn't get too metabolized by your
Mm.
system, so this could be sublingually, but if you take it as a mucosal, so
Oh,
you spray it in your nose,
interesting.
um, is one way to absorb it well, so it doesn't go through your digestive system and get refined.
Mm.
Um, but taking large quantities of it, um, that you can actually get that positive result. And so what I was doing during COVID when I traveled was I made my own nasal spray.
Mm.
And if I went on a plane, um, I use that nasal spray, and maybe it's a little bit, of like airborne, that effervescent, effect, that it's, who knows, but I haven't gotten COVID. So, so
Amazing.
maybe, maybe I'm that person, or not. So, but again, this is where, academic research is really important.
Mhm.
This is where, funding that type of research and doing uh, research that can have peer-reviewed results and publications can lead to our understanding and kind of, scientifically proving uh, efficacy. So
Absolutely. I'm glad that you kind of brought it back to CBD at the end here because that is what we're talking about when we talk about hemp. There's obviously industrial hemp, but that's a different product. Most people who are growing hemp are growing it for CBD and cannabinoids. And there is so much there to learn and explore. And I'm excited for, if we ever get to rescheduling and we can open up these opportunities for more research. And it sounds like the hemp farmers really walk a tightrope between wanting to let the crop grow as long as possible to get the maximum benefit from the CBD and wanting to harvest in time to not have hot hemp and lose all their revenue for that year. So those two things are really in conflict here without some reform.
yeah, and I would be remiss to just talk about CBD. We just do. It's just kind of, just it's the vernacular around hemp.
Mhm.
But there are over 100 cannabinoids and what we call minors. And through
Mm.
our selective breeding, we bred more than 5 million plants during that time period that we did this work. And we found strains and cultivars that were high in, that we could actually breed, that were high in other minors. And a lot of those minor cannabinoids have therapeutic value. And so you can read about CBC and CBG. Others will, put you to sleep, and some will knock out glaucoma and
Mm.
other ailments. So I think there's a lot of value in those minors also, and this is why I don't recommend isolates. And just focusing on CBD. I mean, CBD is amazing, but with inflammation and other applications, maybe with anxiety and a little bit of sleep, but there are lots of minors that we found when if I were to give something to a friend or family member, it would be a full spectrum extract from a hemp plant that even had, 0.5% THC in it, with a warning that if you've never used cannabis, then, just take it slow, because there may be a little bit of a residual effect for people that are sensitive. So.
Mhm. Wow. Such a powerful plant. So many opportunities. Well, thank you so much for asking those questions. As we end here, I just want to ask, what research are you currently working on that you're most excited about that you'd like to share with our audience?
That's not on this topic.
Yeah, no, anything. Just
Well, I mean, I was talking to you right before. We are working on development projects in California in high fire risk areas, and working on innovations that help to mitigate communities from disasters and the environment from disasters. In one of those projects, what we learned is that irrigated agriculture acts as a fuel break for fires.
Makes sense.
So hemp is a good example. The project we're working on, the development up in Lake County, is a community that they want to have a diversified agriculture on site as part of the artisanal, marketing of the project. And I had recommended them growing hemp, and the concern was the downwind odors from the hemp. And there's one of our strains that exhibits terpenes that smells like bubblegum and doesn't smell
Yummy.
like the old bubblegum, the Hubba Bubba stuff. And that's, these are ways that you can incorporate this into our communities without the challenges. So yeah, I think that type of research on how we can tie agriculture, medicinal research, and plants into building resilience in our communities for the future of climate change is important.
Yeah, absolutely. We really need all hands on deck with those types of things. So I very much appreciate you doing that work. Well, thank you, everyone. What a fun discussion. Dr. Azwell, thank you so much for your time, for taking time out of your day to share your research with us and speak with our audience. We've really enjoyed this. Thank you to the audience for participating, and please everyone consider joining us on October 18th for a discussion with Dr. Robert Chilela from UCLA on employment and social equity issues. And we also have a webinar on European cannabis policy coming up on November 1st. You can view our link tree to register and also sign up for our newsletter using the QR codes on the screen. We'll leave it up for a few minutes so you can link as needed. Again, thank you, Dr. Azwell, so much. And to everyone else, have a great weekend, and we'll see you next time. Bye.
Thank you.